So today is the last day of new content for our heredity unit. We might learn about some biotechnology through our review process. Um, but this is the last day of like a traditional information presentation of genetics, and also of like molecular style stuff. We'll start natural selection-y things and ecology after this 9 weeks. So, today we are going to talk about genetic changes that control the cell cycle, because the cell cycle is controlled by genes, and if we disrupt those, it can lead to some pretty bad stuff. So, any questions before we get started? All right. So, first things first, there are specific genes associated with cancer in humans. So, as a little review from our cell cycle stuff, we talked about cancer, we talked about the cell cycle. This is the first slide here. We talked about cancer when we talked about the cell cycle and how there are certain pit stops on that cell cycle that tell the cell to start or stop, right? And if they blow past those checkpoints, that's when you can get cancer. Uh, so the gene regulation systems that go wrong during cancer are the same systems that we've been talking about that play important roles in development, immune systems, and other biological processes. So the systems that we've been talking about that are controlled by these genes are also the systems that are in place to control cell cycle regulation. And so if we disrupt those genes, not only are we disrupting these pathways, but we're also disrupting the cell's idea of when to divide and when not to divide. And when that happens, um, the cell has the ability to become cancerous. So, any questions about this stuff before we get into the technicalities of it all? So, we have control mechanisms in place to control growth. Those control mechanisms are controlled by genes. If we mess up the genes, we mess up that control. If we mess up that control, it can lead to cancer. That's what today is about. It's not necessarily the most fun way to go through a Friday, but it will wrap up everything that we've talked about in a nice little bow. So, so these genes normally regulate cell growth and division during the cell cycle. Include genes for growth factors, receptors, and intracellular molecules. So, genes that regulate cell growth bum, 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 include genes for growth factors, receptors, and intracellular molecules. We don't have to get word for word in here. We'll just get the good stuff, right? The genes that normally regulate the cell growth and division during the cell cycle are genes for growth factors, the receptors, and these intercellular molecules. Mutations that alter any of these genes can lead to cancer. So, if we mess up a cell signaling receptor, if we mess up a cell signaling molecule, if we mess up the actual growth factor itself, or the molecule that it sends into the cell to convey its signal, right? Any disruption in this pathway can cause these changes and throw off the rhythm of the cell cycle. And when that happens, it doesn't always mean cancer, but it very likely could be. So we got some vocab to kind of help clarify what some of these genes are. Vocab that you might have heard before, might not have gotten separated out, and that is oncogenes and proto-oncogenes. So oncogenes are cancer-causing genes. Proto-oncogenes are the normal versions of those genes. So oncogenes have the mutation that can lead to cancer. Proto-oncogenes are the normal versions that code for proteins that regulate cell growth. So proto-oncogenes are genes that control cell growth and division, but have the potential to become oncogenes, and oncogenes are the ones that are mutated and cause cancer. So oncogenes are dangerous. Proto-oncogenes have the ability to be dangerous if mutated in the proper way. We will talk about some specific oncogenes later on in this lesson. So we had a proto-oncogene, if it was mutated, can become an oncogene. This is a cancer-causing mutation. So everybody has proto-oncogenes. Everybody has proto-oncogenes. They are the genes that regulate the cell cycle. If a mutation happens, and that mutation messes up the function of that proto-oncogene in a way that causes cancer, it is now considered an oncogene. So, how do proto-oncogenes become oncogenes? So, here's what the important information is. The title, obviously, that's the question we're trying to answer. Oncogenes arise from genetic change. Okay, so oncogenes arise from a genetic change that leads to an increase in the proto-oncogene product or the activity of it. So, cut out the prepositional phrases, essentially. And these changes fall into three main categories. They are movement of DNA, amplification, or point mutations. So there are three different possibilities for this change to occur. We can move the DNA within the genome. So if a proto-oncogene moves to a new location and has a negative effect, either through increasing in the product or increasing the activity of the protein, right? That will that can change the proto-oncogene into an oncogene. If it amplifies the proto-oncogene, so something happens in the promoter, right? If there is a mutation in the promoter or the enhancer of this proto-oncogene, it can become an oncogene if it's turned on all the time. Or if there is a point mutation in the control system of the proto-oncogene or the proto-oncogene itself. So point mutations, amplification, or movement. Those are the three ways that proto-oncogenes can become oncogenes when we're talking about mechanics. And the next slide is a visualization of that. So this right here is a proto-oncogene, right? If there is a translocation, so the genes moved to a new location and therefore under new controls, we can get our growth stimulating proteins in excess. Excess growth stimulating proteins means more growth, means uncontrolled growth. So if it's moved to a new place, the new promoter could have different regulation systems, and that could be bad. That's one way. Gene amplification. So if we get multiple copies of the gene, right? So if that one gene gets doubled or tripled, then we can get our excess of proteins. If there's a point mutation, right? There's a point mutation in the control and it stops suppressors from binding, right? That could cause more proteins to be transcribed and translated. Or if there's a point mutation within the gene, we can get a hyperactive version of the protein or a protein that can't be degraded. So that means they are in the system longer and more growth proteins and just around more proteins will have like a similar effect. Does that make sense? Okay, these are the ways proto-oncogenes can become oncogenes in a bodily system. We also have tumor suppressor genes. Tumor suppressor genes are genes whose products inhibit cell division. So, tumor suppressor genes have products that inhibit cell division. Since these proteins, yes, they are called tumor suppressor genes since the proteins they encode for help prevent uncontrolled cell growth. If they inhibit normal cell growth, they prevent uncontrolled cell growth. So if a mutation happens in a tumor suppressor gene, right, it decreases the protein and may contribute to the onset of cancer. So mutation that decreases activity may contribute to cancer. So if we decrease the activity of a tumor suppressor gene, right, we will get tumors because the tumors are no longer being suppressed. They call them tumor suppressor genes not because that's their main function, but because their goal is to stop cell growth. And if we don't stop cell growth, that's how we get tumors. Notice that may right there, right? We can get tumors that aren't cancerous. There are multiple steps that you have to take in order to get from a tumor to actual cancer. Okay, I thought I was going to sneeze. So, I don't know whether or not to. I usually try to keep my mask on when I sneeze, but if I have the mic on, I don't want to sneeze directly into the mic. All right, let's just try not to sneeze with this. Any questions about tumor suppressor genes? Okay. So this is kind of where tumor suppressor genes come into play, right? This is review from our talk of the cell cycle. We've got our M phase, G1, G2, right? G1, S. I don't know why G2 and M are switched, but anyway. Oncogenes promote cell growth, right? We get our cell growth from our oncogenes. Tumor suppressor genes will stop that cell from going into G1S, right? I think G2 and M are just switched because it should be G1S, G2M. But oncogenes promote mitosis, right? Allow mitosis to happen. Tumor suppressor genes stop the growth in the first place. They keep it in that resting G0 phase. So if the stop sign goes away, right? If our tumor suppressor gene goes away, then that thing will just keep going and keep going and keep going and keep going, and we don't want that. We're taking away the stop sign that prevents stuff from growing. I've got a couple examples of cell signaling pathways, right? If we interact with cell signaling pathways, that is one of the ways we can cause cancer. And one of specific example is the RAS gene. RAS is a G protein that gives a signal from a growth factor receptor in the plasma membrane and starts a cascade of protein kinases. We've talked about this before, just in very general terms, right? Where we had a cell membrane, we had a receptor, and when that receptor is activated, it will send a cascade of signals and start stuff within the cell. Remember this stuff? All right. So RAS is one of those proteins. RAS would be like right after the signal has met the receptor before the cascade. This is RAS. So the response at the end of this pathway is the synthesis of a protein that stimulates cell cycle. So this stimulates cell cycle. So RAS is a integral part of the system that goes from a external signal to starting the cell cycle. So a cell gets a signal, says, okay, let's start the cell cycle. So they receive that signal. RAS starts the cascade. And at the end of that cascade, we have our normal cell cycle regulation, right? If we disrupt this RAS gene, that's one of the key ways that we lose control of the cell cycle. If this RAS gene has a mutation, stops working, stops functioning, we lose cell cycle regulation. So this is an example of an oncogene or a proto-oncogene that could become an oncogene if it doesn't work properly, because this is a protein, right? This protein has to be synthesized. This is a protein. So there is a gene that leads to this protein. That makes sense? Kind of pushing a bunch of new stuff and old stuff together. So a gene makes this protein. This protein is a part of this signal cascade. If there is a mutation in this gene, it messes up this protein. There's not a functioning RAS. There's no stimulation of the cell cycle. No stimulation of the cell cycle, improper cell growth. Questions about RAS? All right. We also have P53. So P53 would be another example, kind of an opposite example of RAS. P53 is a tumor suppressor gene, and it encodes, sorry, typo, didn't catch that. Is a protein that encodes a specific transcription factor for synthesis of inhibiting cell proteins. So, I'll say this is P53. P53 is transcribed and translated into a protein. That protein So this is the normal pathway. We start with the P53 gene. It is transcribed and translated into this P53 protein. This p53 protein will promote synthesis. So it will bind to our promoters, bind to our enhancers, promote the synthesis of this protein right here. This protein is a cell cycle inhibitor. That cell cycle inhibitor stops cell division from happening. It's a big long pathway just to say stop to the cell. Now, if there is a mutation in p53, that means there's no p53 protein. No p53 protein means no expression of this inhibiting protein. No inhibiting protein, excess division. p53 responds to a lot of things and does a lot of things. If there is nutrient depletion, hypoxia, DNA damage, ribosomal dysfunction, the telomere stuff, or I can't read the specific word, but if telomeres get damaged, right? p53 can be activated. It can lead to cell cycle arrest, apoptosis, DNA repair, specific metabolism stuff. This p53 gene can do a lot of things that either stop division or like nix the cell in general, right? If p53, if this gene gets mutated, none of this stuff down here, proper metabolism, cell cycle arrest, apoptosis, none of that can happen without this p53 gene. I think p53 is referred to as like the guardian angel gene in a lot of cells because it does a lot of stuff to help protect against cancers. So if p53 is mutated or gone or broken or not functioning properly, we have no p53 protein, and without p53 protein, cancer is more and more likely the longer it goes on. So the hope is that if a singular cell has a mutation in the p53 gene, other processes can come apoptose the cell, but if not, then it can cause some pretty damaging effects. Yes. Mhm. There might be, if they do that, it's probably like a clinical trial still in development kind of thing. Gene therapy would probably be the like end goal of something like that, being able to go in and edit that p53 gene to no longer have the mutation so the cell can do what it needs to do. But gene therapy is very, very lucrative at the moment. But future goals, yes. Understanding these processes helps revolutionize medicine every single day. Good question. Anybody else? All right, that's p53. Oh, that's fun. Yes, this is essentially the drawing I just made on the board. Normal p53, right? You have your proteins being synthesized. No p53, no proper or improper transcription. If that makes sense. All right. So, but just because you have one of these mutations does not automatically mean you have cancer, right? There is a multi-step model of what cancer is or what cancer will be. More than one somatic mutation is normally needed to produce all of the full-fledged kind of qualities of cancer. And this is a very, very well supported with the study of colorectal cancer. So that's why I've got this little example in here. Normally, we've got our normal epithelial cells. If we lose a tumor suppressor gene, specifically the APC gene in this case, right, we can get a small benign growth, also known as a polyp. This is why after you reach a certain age, colonoscopies are a regular like preventative medicine care or solution. People with intestinal disorders, celiac disease, not cholestasis. What word am I looking for? Oh, it's right here. Crohn's disease. Oh, I'm gonna think of it in like 20 minutes. So, people with regular intestinal disorders tend to get colonoscopies earlier and more regularly because they have some of these like possible risk factors. So, in colorectal cancer, the first is a very, very small benign growth. Those can be treated very, very easily with regular treatment, and if we screen for it relatively quickly. If we activate a RAS oncogene or we lose a tumor suppressor gene, that polyp can grow. So if this goes untreated or unseen for a prolonged period of time and it hits another one of these mutations, it will continue to grow. Even then, it is still benign and still removable, but it may cause more damage to the intestinal system than it would have if they caught it early. And finally, if we get even more additional mutations like the loss of P53, then we get our malignant carcinoma tumors that are capable of spreading. So this is a multi-step process, and why screening for things like breast cancer with regular mammograms after you reach a certain age, or checkups for prostate cancer or regular colonoscopies are so important. Because if we can catch it with just one or two alterations instead of 3, 4, or 5 alterations, it greatly decreases the risk of that tumor becoming cancerous. Make sense? All right. So, this is where we get into inherited predispositions, right? These are genetic changes that we are talking about, mutations. And some of those mutations, if they happen in the germline, can be passed down from generation to generation. The fact that these genetic changes are required for cancer kind of supports the idea that certain cancers can run in families. So, an individual who gets an oncogene or gets a mutant allele for a tumor suppressor gene is one step closer to getting enough mutations, right, to develop cancer, but not necessarily destined to get cancer. One of the more common genetic predispositions to cancer is with the BRCA gene, specifically BRCA1. We have found that mutations in the BRCA gene are able to be passed down from generation to generation, increasing the likelihood that someone can get breast cancer. A lot of people are tested for BRCA and choose to take action against it preventatively, either by regular checkups, more frequent checkups, or full-fledged mastectomies just to like nip it. So, while these inherited predispositions aren't necessarily you are going to get cancer, it does increase the likelihood that you would. I'm trying to think of a good metaphor. Can't think of one off the top of my head. If I do, I'll let you know. So essentially, if you need like 5 or 6 mutations in a cell in order to develop cancer, you can inherit 1 or 2 of them, and that kind of increases your likelihood because it kind of puts you ahead of the game in the mutations front. So, not destiny, but does increase probabilities. And BRCA is not the only genetic predisposition, but it is one of the more studied. BRCA stands for breast cancer, BR, breast, CA cancer. There are multiple BRCA genes, hence BRCA1, but BRCA1 is the one that is inherited more often. Any questions about this? All right. Genetic predispositions aren't the only way that these mutations can happen. They can also be affected by environmental factors. So, DNA breakage contributes to the likelihood that mutations occur, right? The more DNA is broken, the more likely it is that something gets repeated or lost or mutated. So, the risk of cancer can be lowered by minimizing exposure to some of these things. UV radiation, right? That's why we preach SPF so strongly, because if we can protect our skin and our cells from radiation, we decrease the likelihood that enough mutation would happen that skin cancer would occur. Radon, right, is a radioactive substance, and exposure to it can cause DNA mutations. We talked about Rosalind Franklin, right, and how she was exposed to X-ray radiation every single day at work because she worked with X-ray crystallography, and that it is very likely that that is why she had ovarian cancer. Something I just learned about recently is that a lot of the I don't know what time period it was in, maybe World War II. Don't know. They were in watches. The dials on watches used to be painted with radium. And a lot of women were employed to do this hand painting. And a lot of women got cancer from working with radium all day, every day. And it took a very, very long time for the workforce and scientists to realize that the reason that all of these women were getting sick was exposure to radium. Tobacco chewing and smoking are also really, really big ones, right? I'm sure you've seen the no smoking ads every day since you were like 2 or 3. But mouth tobacco particularly leads to mouth and gum cancer because it is just sitting for prolonged periods of time. Smoking, and now studies are showing vaping can introduce carcinogens into your lungs and cause cancer in your esophagus or lungs themselves. Gamma radiation, asbestos, and many, 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 many more are all things that have the ability to mutate your DNA, and the more mutations you have, the more likely cancer is to occur. So this is not an exhaustive list by any means, but definitely some of the big ones. And finally, viruses. Not viruses like COVID. There are other viruses that are essentially called tumor viruses that can cause cancer in various animals, including humans. These viruses interfere with regulation in a bunch of different ways. So they can integrate their genetic material into the DNA of a cell, which can donate an oncogene, disrupt a tumor suppressor gene, convert a proto-oncogene into an oncogene, all of which are ways that we learned were possible carcinogens, right? Examples of these, the Epstein-Barr virus and HIV-1 are considered viruses that have been linked to increased cancers in the past. So again, just because a person has HIV-1 or Epstein-Barr doesn't mean they will definitely have cancer, but it does increase their likelihood because these viruses have the ability to insert their genome into our genome, and if that happens in the wrong place, it could be very, very bad. That did not take as long as I thought it would. So I don't have an exit ticket for you today, but if there's any other past exit tickets that you haven't done, you can knock those out now. If you have something to do for other classes, you can do that now. We don't have a heck of a lot of time. If you have any questions about this stuff or about anything else we've learned, please let me know. And next week, we'll do some review, talk about biotechnology a little bit, just to kind of support that review, and probably take those progress questions at some point throughout the week. So this will wrap up heredity, and then we will move from heredity into natural selection and evolution, which is really, 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 really fun, especially in AP. So Cool beans.